So as we continue our study of chromosomes, let's talk about gross mutations. So these aren't gross mutations in the sense that they're disgusting. It's the sense of gross as in large. Like you talk about taking gross anatomy as you go into medical school. It's large-based anatomy. You're not looking on a cellular level. So let's look at gross mutations. When we talked about point mutations before, now we're talking about alternations on a chromosome level. So large-scale chromosomal alternations. And this usually doesn't work very well, right? So when you make large changes to the blueprint, usually the building doesn't stand very well. So oftentimes these spontaneously abort. So it doesn't create a viable organism. But there are some that do. So let's consider some of the cases that result in gross mutations, and then we'll look at some of the diseases that we see specifically in humans that are a result of gross mutations or chromosome alteration. So nondisjunction is one of our common sources of gross mutation, where we're going to get the wrong number of chromosomes. So a nondisjunction is we have a mistake during meiosis. So literally the chromosomal pairs don't separate from each other. Or we don't have a separation of two sister chromatids in meiosis II. And so either in one of these cases, when you have a mistake along the metaphase plate like that, you're going to get the wrong number of chromosomes, either one too many or one too few. All right? And when you get one too many or one too few, it's called aneuploidy. So the result of nondisjunction is aneuploidy. So you have the wrong number of chromosomes. You're either monosomic for a particular chromosome, or you're trisomic for a particular chromosome. Now, if you're an even number, if you actually jump ploidy, polyploidy is what you get. So there is normal development in some plant tissues that result in triploid tissue, and then many plants actually are tetraploid, having four sets or four copies of every chromosome, all homologous. Now, it says polyploidy is common in plants but not in animals, and that is true. But when I was taking this class way back, my intro bio class, were I to say, oh, is there ever an instance of animals with polyploidy, the answer would have been no. But of course, never say never. And as we study nature more and discover new things, of course, there's a mammal that actually is tetraploid, and they're discovering more stuff all the time. So this is a rodent they found in South America or Central America that is in fact tetraploid. So we don't want to get too caught up in ultimates. All right. Now, if we alter the chromosome structure, we also can see massive changes. So we have deletions, duplications, inversions, and translocations. Right? So just a diagrammatic look at that. So you have deletion, you remove a part of the chromosome. Duplication, you duplicate a part of the chromosome. Inversion, you flip it. And then reciprocal translocation is you're swapping pieces. Now, if you think back to the karyotypes of the cancer cells that we looked at, this is there's a lot of this action going on in those cancer cells, promoting a cell whose number one goal is to make more of itself. So we do see these. Again, when you mess with the blueprint too much, you usually get something that's not at all what you want, or just doesn't grow at all. Now, let's look at some of the human disorders that are due to chromosomal alteration. So we're going to look at trisomies. First, trisomy 21. This is Down syndrome. We have aneuploidy condition, 3 copies of chromosome 21. This is probably the most common trisomy in humans, and certainly is viable. There's another very common trisomy, trisomy 18, which I've actually had friends who had children with trisomy 18, but they don't survive. But you can do genetic testing for that. So you can see here in the karyotype of this child with Down syndrome, if you look at chromosome number 21, there are 3 of them. So this individual actually has 47 total chromosomes instead of 46. Now, there's aneuploidy of the sex chromosomes as well, and these have less dramatic effects. There's Klinefelter syndrome if you're XXY. The XYY males are also relatively normal development, but they tend to be a little bit taller. XXX females, again, tend to be a little bit taller. And then Turner syndrome is named for XO females. And so this is an individual that is sterile, but it's the only known viable monosomy in humans. Right, so this individual actually has 45 chromosomes only instead of 46. Right? Now, when you start to alter chromosomes, there's a few syndromes that you may come across, again, in your studies of biology or medicine. One is called the cry of the cat. It's a very specific deletion in chromosome number 5. These individuals usually die very early. And then CML, chronic myelogenous leukemia, is caused by a translocation in chromosomes 9 and 22. And so, in fact, the Philadelphia chromosome is named as such. It's distinctive enough and it's common enough in this genetic disorder that it has its own specific name. So again, there's not a whole lot of these, but I do want you to know that they can happen and what the results are.